All right, we looked at them a little bit in the last video, but here's just a little definition of the two different types of modeling when we draw out atoms. So the first one's a space-filling model. This one's good because it shows us the atomic radius. We draw the particular element based upon its size, and so that helps us kind of visualize that idea, gives us a better idea of Coulomb's law. It also shows us then as a result the relative bond length, but however it doesn't really show us the three-dimensional positions, and it does not give us the number of bonds because double, single, triple, they're all just crammed together and pushed into one. The ball and stick model, so it does a better job of giving us three-dimensional positions. It gives us the single, the double, the triple bonds. However, the balls are not proportional, and so does not give us a clear picture of the bond atomic size. So therefore, Coulomb's law, it doesn't really help us out that much. And the sticks aren't proportional to the bond length, so single, double, triple, the lines are always drawn the same length. Resonance structures. So this is another problem that you're going to run into with Lewis structures. Sometimes you're going to find that the double or the triple bond can be located on different atoms. And this isn't a question of the formal charge. Instead, this is just how you draw it. And there might be multiple ways to draw it that mean the exact same thing. So this can result in two or more possible Lewis diagrams that are equally valid. They would have the exact same formal charges, and so picking the right one, well, they're all the right one as it turns out. So for example, here's ozone, O3. If we were to draw out the Lewis structure and figure out the formal charge, we'd find that both of these work. The difference between these two, that one has the double bond there, that one has the double bond there. So these are ball and stick models, so we can't really see the relative length. But remember, these bonds right here are going to be shorter than that bond right there because they're double bonds, and so you're going to have a short bond and then a longer bond. Now you see this little back and forth arrow right there? That means this ozone molecule with its double bond on the left-hand side is the same as that one right there because that double bond is actually going to flop back and forth. It's not locked into this particular shape. Those two electrons might go over to this side, and so it's going to keep cycling back and forth constantly. So within a sample of ozone, there's going to be billions upon trillions of ozone molecules. Each one of those individual ozone molecules, its double bonds are going to be flipping back and forth. That's why it's called a resonance, because it's constantly moving. And so as a result, this bond is shorter, that bond's longer, but then it's going to jump over there. That one's going to become shorter, that one's going to become longer. So normally there would be 1, 2, 3 bonds here, but since it's moving around and it's spending a different amount of time with each guy, the overall effective number of bonds is going to be 1.5. So what this means then is on average, since those electrons are spinning around, usually there's going to be 3 electrons on either side in total. So the average bond number would be 1.5 for these guys. Here's nitrate. So nitrate has a double bond between nitrogen and oxygen, and all the oxygens are the same. The only difference is where that double bond is located, and it can be found on either of those oxygens. And so as a result, there's going to be 3 different versions of nitrate, and one isn't going to stay that version. It's going to keep hopping back and forth. So the effective number of bonds for nitrate, there is a total of 1, 2, 3, 4 bonds. They're jumping around between 3 atoms, so that means the effective bonds are 1.33. And so then the bond length, the effective bond length for all 3 of those, would be a little bit more than a single bond and a little bit less than a double bond. It's going to have the bond length of 1.33 bonds. This is a very common AP question. So make sure you know that. Count the number of bonds, divide by the number of atoms. All right, here's another one. On this guy, it doesn't just have 2 types of atoms. We have 2 oxygens bonded to carbon, and then we also have a hydrogen. So that double bond is not going to jump over here to hydrogen because hydrogen cannot have double bonds. What we're going to have here then is just 1, 2, 3 bonds attached to 3 atoms. I'm sorry, 2 atoms, the 2 oxygens. And so that means the effective number of bonds is going to be 1.5. So the bond length then for these bonds would be approximately a 1.5 bond, whereas this guy, it's going to be, a single bond. So full-blown single bond and then a 1.5 bond. So these bonds are going to be longer than this one right here. So if we look at nitrite and nitrate, NO2 and NO3, question is which structure has the shortest bonds? Now, if you don't know anything about resonance structures, you would say, well, this guy has got single bonds and that one's got double bonds. This guy has single bonds and that one has double bonds. So they're the same. But that's not true based on what we know now. So right here we have 1, 2, 3 bonds, and we have 2 atoms, that means the effective number of bonds for this fella is going to be 1.5. The nitrate down there, we've done that one already, and it winds up that it has the effective number of bonds of 1.33. So shorter bonds, remember, as we get higher, more bonds, the bonds get shorter. So a single bond is the longest, triple bond is the shortest. That means with 1.5 bonds, like we have right here, those bonds are going to be the shorter ones. So resonance is important because of those effective number of bonds. If we were to do something like nitrate or look at nitrate and try and calculate in laboratory tests, figure out the bond energy and bond length, it wouldn't come out like we expected it. We wouldn't find that nitrate has 2 single bonds and a double bond. We would find that overall it has 1.33 bonds. And so this is really important in justifying the data that we get inside of a laboratory.